This is the Chapter 13 Carbohydrate Metabolism Part 2 commentary. In this commentary, we're going to be talking about the breakdown or catabolism, as well as the buildup, anabolism, of carbohydrates. To start us off, the next several slides may seem complicated, so I've tried to put together an even more complicated summary diagram. So let's go ahead and focus on this part right here. I'm going to go ahead and highlight this circular. Again, this is just a summary diagram of the key concepts for the remaining slides. So in this right here, we have at the very top glycogen. That's a polysaccharide which the body uses as temporary storage. Now, the glycogen can be broken down into glucose. So polysaccharides into individual monosaccharide glucose molecules, which can readily be used for energy. The name for the breakdown of glycogen to glucose is called glycogenolysis. Glycogenolysis. You can see where they get the name glycogen, and then they add lysis to separate. Now, when we're actually breaking down glucose in order to release energy to regenerate NADH or NAD to NADH and ADP to ATP, going from glucose to pyruvate, we call this glycolysis, which we've already talked about. So this is the catabolism, which I've went ahead and highlighted using a red marker. Let's go the opposite way. Now it turns out we can go ahead and take pyruvate molecules and convert them, spending energy, but convert them back into glucose. So we call this Gluco Neo Genesis. So the building up of new glucose molecules. We can then take those glucose molecules, if we already have too many, we can go ahead and turn them into a temporary storage form, going from monosaccharide to a polysaccharide storage unit. And going from glucose to glycogen, we call glycogenesis. So, let's go ahead and I'm going to make reference to this summary diagram as I go through the next part. So when we're building up our glycogen, from glucose to glycogen, again we call this glycogenesis. Glycogenesis. So glucose to glycogen is glycogenesis. So that's for the next slide. It requires a unique type of battery called UTP. And UTP gives off a phosphate in order to power up the linkages of the glycosidic linkages of the glucoses to form glycogen. So it gives it up, forming UDP. So think of UTP as just like ATP, where it provides the energy in order to build up the glycogen. Glycogen is mainly stored in the liver and the muscle. I should point out that the liver glycogen, this is going to be later distributed throughout the body. So even though we're temporarily storing the glycogen in the liver will then go ahead and break it down at a later point and distribute it out throughout, and I know it's kind of messy, throughout the body. Glycogen, which is stored in the muscle, it basically stays in the muscle, and the glycogen is broken down for energy exclusively for the muscle. So those are key differences between the glycogen found in the liver and glycogen found in the muscle. And when you have an excess amount of glucose, glycogenesis occurs, and the body naturally tries to store up both of them at the same time, both in the liver and the muscle. The next one is glycogen breakdown into glucose. It's not written on here, but again, the name is Glycogenolysis, with lysis meaning breaking down. So if we went back to slide 10, it's this part right here, breaking down glycogen to glucose. So, in order to first break it down, we need to go ahead and prime it or power it up so it can be released, or so it can be broken down later to be released for energy. So it seems kind of weird adding on the phosphate because we are actually wasting energy. Let me go back to my analogy here. So now let's go ahead and show you a white screen. Uh-oh. There we go. Sorry, technical difficulties. Here is a potential energy diagram. Here is a boulder that's stuck. So what we need to do is we need to provide a little bit of power or push in order to bring it over the hump of the energy curve, at which point then it starts crashing down, running into people. Oh no. So in this instance here, we went ahead and we added in a fast phosphate, wasting a little bit of energy in order to get it primed or powered up. Now, in this form right here, it will then be mutated from glucose 1 phosphate to glucose 6 phosphate. And again, it's primed or powered up, ready to be broken down for energy. The body has a couple choices. It can go ahead and remove that phosphate so that it's just a generic glucose. And this is the form that you would use and transport it in blood. So this is blood glucose, for example, or this is just generic glucose. This glucose over here, glucose 6-phosphate, this one is already primed and ready to go, so this could be directly inserted into glycolysis in order to be broken down. So, I bring this up because it turns out that in order to go from glucose 1 phosphate to glucose 6-phosphate, as well as go to glucose 6-phosphate to glucose, you need the proper enzymes. The first enzyme for glucose 1-phosphate to glucose 6-phosphate is called Phosphoglucomutase. And when it comes to the liver and muscles, they both have these. However, when it comes to this form right here, glucose 6-phosphate, and let's say we wanted to turn it into glucose, it's only liver that has this particular enzyme, only the liver. See, the reason liver only has this particular enzyme because the liver may change its mind and may want to turn this glucose back into glycogen at a later point. Unfortunately, muscle cells do not have this enzyme. So keep in mind that muscles have their own glycogen storage. So when muscles break down the glycogen, automatically it's in this charged form. So the good news is it's ready to be broken down immediately via glycolysis for energy, which is awesome because that's what muscles do. They need to go ahead and break down the glucose, or break down the glucose immediately, so that way they can release energy. On the other hand, they do not have that enzyme, so they can't sit there and remake their glycogen. That's the liver's job. So they're very elitist. Only the liver can sit there and make the glycogen. So they're great at at burning energy, but muscle cells are not very good at making energy or storing energy. So let's go ahead and move on. I beat that to death. Gluconeogenesis. So this is the synthesis of new glucose from non-carbohydrate molecules. So when we start running low on glucose, and there are certain parts of the body, such as the brain, that need constant fresh supplies of glucose, then what the body will do is it will start looking for biomolecular spare parts in order to build glucose. So we call this gluconeogenesis. If we go back to my slides, so in this case right here, I actually showed you pyruvate, and it's converted back to glucose via gluconeogenesis. However, you can actually use other molecules, lactate, which we'll get into a bit. You can also use amino acids, spare amino acids, in order to turn it back into glucose. So when you start to run low on energy because you haven't eaten for a while and you're starting to starve, then this is what the body does. This can be a bad thing because amino acids can be gotten from muscle or other vital parts of your body. So your body may end up stripping amino acids in order to make this new glucose, so that way it can supply the critical areas. So that's called gluconeogenesis. This pathway mostly occurs in the liver, and very little in other parts of the body. So unfortunately it doesn't occur in the brain, or it occurs very little in the brain. So the liver has the big job of making all of the glucose so it can be transported to the brain. So we talked about amino acids being converted into glucose along with spare parts such as glycerol. Keep in mind that brain and heart do need fresh supplies of glucose, so if you're starting to starve, again, then the body will start to strip other less crucial biomolecules in order to supply enough amino acids, glycerol, and carbohydrate derivatives in order to make glucose. Now, the Quarry cycle and exercise. So let's go back to my picture, and I'm going to go ahead and use my highlighter. In order to understand the Quarry cycle, let's first understand the difference between aerobic and anaerobic. Aerobic, which I went ahead and wrote in purple, look for the asterisks as well as the purple arrows. In the aerobic respiration, you have plenty of oxygen. So high amounts of oxygen. Matter of fact, I'm going to write that right over here. So you have high amounts of oxygen. So in this scenario, glucose is broken down into pyruvate via glycolysis, and then the pyruvate is broken down into acetyl-CoA, which is inserted into the Krebs cycle. And then we've talked about this in the past, which regenerates the NADH and FADH2, which is transported into the electron transport chain, and then oxidative phosphorylation, regenerating the ATP. Bottom line is you need oxygen to do this, so we call this aerobic respiration. And this is the main energy process to regenerate our ATP from ADP so that we can survive. Now, during exercise, you're not getting enough oxygen. So, we have what we call anaerobic respiration. An means not. So this means you have low amounts of oxygen. Consequently, here is the crude pathway. You start off with glycolysis, so you go from glucose to pyruvate. Notice I went ahead and made it green. I gave you green arrows. Actually, in this instance, anaerobic respiration is nothing more than asterisks. So let's go ahead and focus on the green asterisks for right now. So in anaerobic respiration, you go from glucose straight to pyruvate. The problem, however, is since you don't have enough oxygen, then the Krebs cycle slows down. So this is not a immediate answer for getting energy. You can't go the aerobic route. So we're stuck in a dilemma. We're in pyruvate, but we can't form acetyl-CoA. So what the body does is it temporarily breaks down the pyruvate for energy into lactate. And that's how it temporarily gets its energy. Keep in mind this is very temporary. The lactate is then has to be at a later time regenerated back into pyruvate. Otherwise the lactate starts to build up. Now, once it's in pyruvate, the body will go ahead and convert it back into glucose via gluconeogenesis, and then the same process occurs. It's broken back down into pyruvate, lactate, and then it's regenerated back into pyruvate and so forth. This is called the Quarry cycle. The Quarry cycle. So let's go ahead and move forward. Here's a nice picture right here. Here we see active skeletal muscle. This is anaerobic respiration, low oxygen. So in this case, glucose is going to go to pyruvate, and then pyruvate, because there's low oxygen, is going to be converted into lactate to release just a little bit of energy. The lactate, however, needs to be converted back into pyruvate. Otherwise, it starts to build up. And that's when you start to feel exhausted, fatigued, and your body's really struggling for breath. So the lactate literally transfers through the blood to the liver. It's the liver that converts the lactate back into pyruvate and then back into glucose. Once it's converted back into glucose, then the glucose has to make the long journey via the blood back into the muscle. Now, this is a very inefficient process. It's the best solution to a worst situation. So, eventually, it's a losing battle, and you'll have high amounts of lactate build up. And eventually, you'll just simply have to slow down and get more oxygen, which eventually the lactate will then start to diminish, and you'll go back to your normal aerobic respiration. So make sure you know all of these steps for the upcoming tests. So all of this should be put on your cheat sheet. Now, here is hormonal control of carbohydrate metabolism. This is the last part. Now, though there are 3 hormones we're going to be talking about, I'm first going to focus on the first 2, insulin and glucagon. So, insulin is given off to decrease blood glucose levels. Consequently, as it decreases blood glucose levels, it actually increases the rate of glycogen. So, as the blood glucose goes down, the glycogen goes up. Glucagon hormones do the opposite. So they decrease glycogen levels, but increase blood glucose. Now, the reason I'm focusing on the first 2 is due to the fact that the glycogen we're referring to is found in the liver. So, the first 2 hormones really have to do with the relationship between the liver and your blood. So again, insulin will sit there and decrease the glucose which is found in your blood because it transports it and synthesizes the glucose into glycogen, which is found in your liver. On the other hand, if you have a large amount of glucogen which is released, then the opposite occurs. As the glycogen is degraded, then the liver sends it directly to the blood so that your blood glucose is increased, which at that point the glucose basically travels to whatever cells in the body needs the glucose. Now, if you go to the next slide, I like this picture because it shows their relationships. As you increase the amount of glucagon hormones, then the glycogen level, which is stored in your liver, is going to decrease. Consequently, it's going to increase the amount of blood glucose or blood sugar level. On the other hand, if you increase the amount of insulin, it's going to go ahead and decrease the amount of blood sugar, which increases the amount of glucogen. When you have type 2 diabetes, due to various effects, and we won't go into those, but one of the effects is that you're not producing enough insulin when you're supposed to. Consequently, then you end up having really high levels of blood sugar. And it turns out any excess biomolecules in your blood is a bad thing because it causes a lot of unwanted side reactions. So oftentimes people who have type 2 diabetes, as well as a traditional type 1 diabetes, they have to take insulin shots in order to lower their blood glucose level, forcing the liver to turn it into glycogen, which is stored in the body. Now, if you notice, the very last one I've left out is epinephrine. Now, yes, there are more hormones than this, but we're just going to focus on these three right here. And the last one is epinephrine. Now, what this does is it goes ahead and it increases the blood glucose levels. How does it do that? Well, it stimulates the glycogen breakdown from the muscles. So, the other one was glucogen from the liver, but epinephrine stimulates blood glucose levels from the muscle. So, decreases the glycogen, which is found in the muscle, to increase the blood glucose levels. This is important because muscles, again, have their own separate glycogen storage, which is separate from the liver glycogen storage. So, and we like to break down this muscle glycogen storage because the glucose is then readily available to be used whenever there's brief, intense exercise, such as when you're running away from a stranger or fighting or panicking over an exam. You're responsible for all of these hormones and their overall responses on the final, so make sure this goes on your cheat sheet. This concludes the last of the slides. Again, everything should go on your cheat sheet. I recommend summarizing it in your own words, but everything